Hello, this is Interview, and I'm Caitlin LeBrun, your host, and today we're starring Marin Carr.
I'm Erin Carr, the wonderful scientist. Nice to meet you.
It's nice to meet you too. Okay, I have a question for you.
All right, let's hear it.
If two sets of identical twins married and each has a child, could the resulting children be identical?
The odds of having identical twins are 3 in every 1,000 births. The odds of having an identical twin marry another identical twin and them giving birth to identical twins are 1,000,000 to 1. So no, there's no chance that the twins could be identical.
Why is that?
The only way to get an identical offspring is when the fertilized egg splits into two cells.
Oh,
The children wouldn't inherit identical traits from their parents because of a process called genetic recombination.
wow, that's a big word. What is genetic recombination exactly?
Genetic recombination is something that occurs during a special type of cell division that forms egg or sperm cells when chromosomes swap their genes in a unique way, forming genetically unique sperms.
How does this relate to the children not being identical?
Well, because of the process, genetic recombination, all of the female eggs will be different from all of the others. Here's a visual. These are the non-sister chromatids, and this is the first site of them crossing over. You see the variation here, and then you see the tetrads.
That's cool. Okay. Oh, I understand. You're saying that the chromosomes get mixed up.
Exactly. You're starting to get it.
Is it true that identical genes are not identical?
Yes, in fact, that is another reason the children would not be identical.
Really?
Yes, meiosis causes the daughter cells to be different. Identical twins develop from a single fertilized egg. So many differences between the twins are due to their environments, not genetics.
So twins have largely been attributed to environmental influences?
Yes, it has been shown some sets of identical twins differences can spring from unique changes in what are known as epigenetic factors.
What are epigenetic factors?
Epigenetic factors are chemical markers that attach to genes and affect how they are expressed, in some cases by slowly or shutting off or shutting genes off, and in others by increasing their output.
This is what affects genes?
Kinda. This is a visual of epigenetic. These what are the factors that are affected: development, environmental chemicals, drugs, aging, and diet. The DNA methylation methyl methyl group and epigenetic factors found in some dietary sources can tag DNA and activate or repress genes. Histones are proteins around which DNA can wind for compaction and gene regulation. Histone modification is the binding of epigenetic factors to histone tails after the extent to which DNA is wrapped around histones and the availability of genes in the DNA to be activated.
Oh, I understand now.
The specific changes that were identified, known as number variations in which genes exist in multiple copies. Not known, however, is whether these changes in identical twins occur at the embryon's level as the twins age.
So that's the one question you don't know the answer to.
That's not funny. Last but not least, the children of the two sets of twins will not be identical.
Here we have it, folks. Identical twins do not have identical DNA. Twins have been attributed to environmental influences.
That is it. That answers your question.
Thank you.
Good night, everybody.
Good night. See you next time on Interview.